Hello, my name is Joshua Mugabi. I welcome you to membrane structure. Lesson objectives. We shall look at the cell membranes, phospholipid bilayer, membrane models, membrane proteins, cholesterol, membrane diagram, and the cell wall. Let's start with the cell membranes. Cell membrane, these ones enclose the content of a cell. They enclose the contents of a cell, separating the intracellular components from the outside environment. That is what we refer to as the cell membrane, the structure that encloses the contents of a cell, separating the intracellular components from the outside environment. It allows for the control of internal cellular conditions or homeostasis. Cell membranes have two key qualities. One, semi-permeability. This means that some materials cannot cross without assistance. Semi-permeability, some of the materials cannot cross without assistance. And then selectivity. In selectivity, the cell regulates the passage of certain materials. The cell regulates the passage of certain materials. Those are the two key qualities of the cell membranes. Semi-permeability, some materials cannot cross without assistance, and selectivity, whereby the cells regulate the passage of certain materials. Phospholipid bilayers. Cell membranes are composed of primarily of the phospholipids and the proteins. They have the phospholipids and proteins. If you look at the structure of the phospholipid, it consists of the hydrophobic head, hydrophilic head, and then it also has the hydrophobic tail. These ones are the hydrophobic tails. And the one we have on top is the hydrophilic head. This one, the hydrophilic head, is attracted to water, and the hydrophobic tail is repelled by water. The head is hydrophilic, meaning that it is water-loving. That is the head. It is hydrophilic, water-loving. Whereas the tail is hydrophobic. The tail is hydrophobic, meaning that it repels water. The arrangement in the membrane. The membrane, we say that it has got the phospholipid, and the phospholipids, these ones, they form a spontaneous bilayer. So we have the first layer up, and then we have the second layer down. So those are the ones that we are calling the bilayer. The upper part is made up of the phosphates, and the inner part is made up of the lipids. So that's why we refer to them as phospholipids. And there are two layers called bilayer. The tails, they face inwards, and the head faces outward. So what we are having this side, this is the head, which is made up of the phosphates. And what we have inside, these are the ones that we are referring to as the tail, which are facing inside, and they are made up of the lipids. So the outer part, or the extracellular, is what we are referring to as the interstitial fluid. This outer part here is where we have our interstitial fluid. So meaning that the inner part is now what we are referring to as the cytosolic fluid, the interior. In the interior, we have the cytoplasm, and the exterior, or the extracellular part of the cell. It's where we have the outer part of the cell. So the tails are the ones that are facing inwards, and the heads, they are the ones that are facing outward. The phospholipids form a bilayer in water due to the amphipathetic properties of the phospholipid molecules. So we are saying outside here we have our phosphates, which are hydrophilic. They are water loving. So they will be facing the water side. Up on the extracellular side, and inside the cell on the intracellular side. The phosphate, or the heads, are the ones that are facing towards the water. Then in the middle, it's where we have our tails. The tails face away from the water because they are water repellent. And that one is what we are referring to as the amphipathic property of the phospholipid molecules. So the phospholipid form a bilayer in water due to amphipathetic properties of the phospholipid molecules. Still on the properties of bilayers, you will see that the bilayer is held by a weak hydrophobic association between the fatty acids. The bilayer is held together by the weak hydrophobic association between the tail. So if you look at these tails here, they are close to each other, and in between them there is a weak association between those fatty acid tails, and that one is what we are calling the weak hydrophobic association. And it is that weak hydrophobic association that holds the bilayer together. This one up, you can see that the bilayers are fixed together, and it is saturated. It is saturated because the bilayers or the fatty acid tails are held together. But when you look at these ones down, you will see they are forming what we call kinks. They are separating each other. Those kinks are brought about by having double bonds in the fatty acid tails, which makes the bilayer unsaturated. So it becomes a fluid-like. Whereas the upper layer is saturated because all the areas are occupied. They don't have double bonds in the fatty acid tails. These ones are unsaturated because of the structure of the fatty acid tails forming bends, or what we call kinks, which makes it to become unsaturated, and therefore it affects its structure to be a fluid. The structure of the fatty acid tails affects the fluidity. The structure of the fatty acid tails is going to affect the fluidity. Now, for the saturated fatty acid, this one increases the viscosity. And for the unsaturated tails with kinks, they increase fluidity. So here, in the saturated fatty acid tails, the tails are holding each other strongly with the hydrophobic weak force, because the fatty acids are saturated, and therefore it makes it viscous. Whereas here, these fatty acid tails are unsaturated, and therefore they form what we call kinks. And these ones increases the fluidity of the bilayer molecule or of our membrane. So it becomes more or less like a fluid because of the unsaturated tails. Now we look at the Davison-Daniell model. Now, when viewed via an electron microscope, a cell membrane exhibits a trilaminar or three-layer appearance with the two dark outer layers and a lighter layer inside. So this is the model which was proposed by Davison and Daniell. For them, they were saying that the cell membrane shows a three-layer appearance. It has two dark layers, these ones that we are seeing outside, two dark layers, and then he was saying that it has also got an inner layer which is lighter, the one that we are seeing in the middle. So those were the three layers for them they proposed. Davison and Daniell proposed a model whereby two layers of proteins, which are the dark part that we have seen somewhere here, the dark part, the outer part and the lower part, that were flanking a central phospholipid layer, the one which we are looking at as a lighter part in the middle. That's what they proposed, that the cell membrane is made up of two proteins which are dark outside, flanking a central phospholipid bilayer, which is the one light in the middle. The lipoprotein sandwich structure, or the lipoprotein sandwich model. For them, there was a falsification of Davison and Daniell's model, where they say membranes don't have a constant lipid protein ratio. Membranes don't have a constant lipid-protein ratio. This one precludes the sandwich model. So that was a falsification of Davison and Daniel's model, where they say that the membrane does not have a constant lipid-protein ratio. So it prevents their sandwich model. They say that membrane proteins, they vary in size and solubility in the water. Therefore, they could not form an outer layer. That is still falsification of Davison and Daniel's model. These proteins that we find in the cell membrane, they vary in size and solubility in water. Therefore, they cannot form an outer layer. Then still they continue to say, still about falsification, that proteins are said to be more mobile within the membrane, and they are not fixed in a place when tagged with fluorescent antibodies. So it was proved that the proteins were not fixed in one particular point in the cell membrane. They were always mobile within the membrane. They were not fixed when they were tagged with fluorescent antibodies. Hence could not form static outer layer. They could not form a static outer layer as proposed by Davison and Daniel's model. So freeze fracturing, the freeze fracturing of the membrane reveals a rough, irregular internal surface suggestive of a transmembrane protein. By carrying out freeze fracturing of the membrane, it revealed that there was a rough, irregular internal surface suggestive of the transmembrane proteins. So which one we are saying that proteins are not localized on the outer layer. So they were just irregular, roughly, and internal surface moving in different points. So this one falsified Davison and Daniel's model. Analysis of falsification of Davison and Daniel's model led to Singer and Nicholson model. After falsifying the Davison and Daniel's model, it led to a Singer-Nicholson model. Singer-Nicholson model. So we were saying, because of the falsification of Davison and Daniel model, it led to a proposal of a new model. And in this model, the Singer-Nicholson model, it describes the proteins being embedded within the phospholipid bilayer instead of existing as a distinctive outer layer, like the way Davison-Daniel model say that the proteins, those ones on top, they form their own layer on top and also on the lower bottom. Therefore, the bilipid layer were enclosed in between the proteins. But when it was falsified, Singer and Nicholson, they brought up a new model which said that the proteins are embedded within the phospholipid bilayer. They are not on top. They are transmembrane. So when you look at them, you will see that the proteins are embedded within. They are embedded within the phospholipid bilayer, not forming a layer on the top of the bilayer. So that was the new proposed cell membrane structure by Singer and Nicholson. Membrane proteins. Membrane proteins serve a variety of key functions. 1. Their junction. Tight junctions. If you look at this membrane, these proteins, they form what we are referring to as the junctions. They are junctions in the membrane. 2. They are enzymes. They are localized activities that take place in those particular area. So they act as enzymes. 3. They act as transport medium for passage, both active and passive. If you look at them, they are transmembrane. They go through from up to down, which means that they are used for transportation of materials from one side to another side. Therefore, recognition, that is, they are still junctions. They can be used as recognition sites, recognizing other materials. They are for anchorage or attachment sites. Molecules that are moving, they will attach themselves on the proteins in the membrane. They are for transduction. Those are peptide hormones. Types of membrane proteins. We can say that membrane proteins can be categorized either as integral or peripheral. They are categorized as either integral or peripheral. So when you look at them, we have a protein which is transmembrane. That one we are referring to it as integral. And we have another protein which is outside the membrane, which we are referring to as a peripheral protein. So we are saying that the membrane proteins can be categorized as integral or they can be peripheral. Integral proteins. These are permanently attached. They are permanently attached to the plasma membrane, like the way we are seeing it here. This is an integral protein. It is permanently attached to the plasma membrane. That's what we are referring to as the integral protein. Membrane proteins, we have already seen, they are categorized as integral or peripheral proteins. Now, integral proteins, we have seen, they are permanently attached to the plasma membrane. Peripheral proteins. Peripheral proteins. Now, the peripheral proteins, these ones are temporarily associated with the membrane perifence. They are temporarily associated with the membrane perifence. Those are the peripheral proteins. Peripheral proteins. For them, they are temporarily associated with the membrane perifence. They are outside of the membrane. They are not inside the membrane like the integral protein. Those are the peripheral membrane proteins. And can be removed by either the salts or pH changes. Those ones are the peripheral proteins. Because they are not associated with the membrane directly, they can easily be removed by salt and pH changes. Membrane proteins, they are diverse in terms of the structure and position in the membrane and function. Those are the membrane proteins. They differ in terms of their structure, their position in the membrane, and also in their functions. We have the cholesterol. Cholesterol is a component of animal cell membranes, and this is where it functions. Its function is to maintain the integrity and mechanical stability of the membrane. That is the cholesterol. It is a component of a cell membrane where its functions are to maintain the integrity and mechanical stability of the membrane. If it is absent, we find it is absent in plant cells membrane, which are supported by rigid cell wall. So you find that the cholesterol is absent in the plant cell membrane, and these ones are supported by the rigid cell wall. Cholesterol is an amphipathetic molecule. That is, it has got both polar and non-polar regions. It is amphipathetic because it has got both polar and non-polar molecule. So it has got the hydroxyl, this hydroxyl group. That hydroxyl group is hydrophilic, meaning that it is water-loving, the hydrophilic group. And the rest of the molecule is hydrophobic. So this other remaining molecule of the cholesterol, this side, is hydrophobic, meaning that it is water-hating or water-repelling structure. What are the role of the cholesterol? Cholesterol interacts with the fatty acid tails. It interacts with the fatty acid tails, and it modulates the membrane's properties. It interacts with the fatty acid tails, and it modulates the properties. So here we can see this is our cholesterol. It is interacting with the fatty acid tails. You can see it is located inside the fatty acid tails. And being located in that area, it modulates the properties of the membrane. Cholesterol in mammalian membrane reduces the membrane fluidity and permeability to some solute. In animal membranes, the cholesterol reduces the membrane fluidity and permeability to some solutes. So it is going to reduce the membrane being fluid, and then it will also reduce the permeability of some of the solute, meaning that it will control the passage of some solute through the cell membrane. And then it will also control the membrane being fluid in form. It immobilizes the outer surfaces, that is still cholesterol. it immobilizes the outer surface of the membrane, reducing its fluidity. So when it is placed inside the phospholipids, when it is placed inside the tails, it prevents the formation of kinks because it squeezes itself between the tails. And when it squeezes itself between the tails, it will prevent those kinks. And when it prevents those kinks, that means that now it is going to reduce the fluidity of the membrane. It will reduce the fluidity of the membrane. And it makes the membrane less permeable to smaller water-soluble molecules. Because we are having our cholesterol embedded in the tails of the phospholipid and reducing the fluidity of the membrane. This one makes the membrane to become less permeable to all the small water-soluble molecules. And it separates the fatty acid tails to prevent crystallization at low temperature. So, the presence of the cholesterol in the fatty acid tails is going to prevent them from crystallization at lower temperatures. It helps to secure peripheral proteins by forming a high-density lipid raft. We are saying it secures the peripheral proteins by forming a high-density lipid rafts. It secures the peripheral proteins, supporting the peripheral proteins by forming a high-density lipid rafts. Let's look at the fluid mosaic model. This is the drawing of the fluid mosaic model. When you look at it, we have the integral protein, transmembrane. It moves across the bilipid layers, phospholipid layers, that is the integral protein. Then we have the cholesterol. The cholesterol is the one that we are seeing embedding itself in the tails. That one is the cholesterol. Then we have the glycoprotein. Glycoprotein are on the surface of the membrane, form the glycoprotein, which is made up of carbohydrates and protein. Then we have the phospholipid. Phospholipid, the outer part, is what forms what we call the phosphate. And then the tails, down parts, are what we call the fatty acids. The lower parts are what we call the fatty acid. Those ones, they form what we call the phospholipids. And then we have the peripheral proteins. Peripheral proteins, these are the proteins that are attached on the surface. They don't go deep inside the phospholipid bilayer. Those are the peripheral proteins. Those are the different parts that we find in a fluid mosaic model. Integral proteins, cholesterol, glycoproteins, phosphate and fatty acid, which form a phospholipid, and a peripheral protein. Then we have the phospholipid. Let's look at the cell wall. A cell wall is an outer layer which is surrounding some types of cells that provide additional structure support and protection. That is a cell wall. It is an outer layer surrounding some types of cells that provide additional structure support and protection. If you look at our stained cell wall, you can see that you have the cell wall there. Those structures there, they are our cell wall. Surrounding, moving around, you have, that is a cell wall. Those ones are the ones that we are saying they are outer layers which surround some types of cell to provide additional structure support and protection. They are what we are referring to as the cell wall. They are important controlling the hydrostatic pressure. They are important in controlling hydrostatic pressure. The cell wall may be composed of cellulose, that is in plant cells. It can be composed of peptidoglycan, that is in bacteria, and chitin in fungal cells. Those are the cell walls. Either it's composed of cellulose, like in plant cells, peptidoglycan in bacterial cells, or chitin in fungal cells. The plants and algae cells may be connected by small channels. Those small channels, we refer to them as plasmodesmata. And they are extant between cell wall. They are an extension between the cell wall. That is in plants and algae cell. That's where we can have some channels connecting one cell to another. Like you can have a channel here, that channel there, another channel there, connecting the nearby cells. Those channels are the ones that we are referring to as the plasmodesmata. So, we have been looking at the membrane structure, and our objectives were the cell membrane, phospholipid bilayer, membrane models, membrane proteins, cholesterol, membrane diagram, and the cell wall. Thank you. Don't forget to subscribe.